everybody. Welcome to OsteoBites. Thanks so much for joining us today. We're so glad you made the time. We'll give everyone a few seconds to get settled in our virtual room. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and Director of Scientific Programs at MIT Agents. And today on OsteoBites, we are joined by Dr. Fan Yang, an associate professor at Stanford University, and she will be discussing her work on tissue engineering strategies for elucidating osteosarcoma biology and drug discovery. Thank you so much, Dr. Yang, for joining us on OsteoBites today. We are thrilled to have you. And thank you also to Walker, our panelist today, for joining us. A little bit more about our guest today. Dr. Yang is an associate professor with tenure at Stanford University with joint appointments in the departments of orthopedic surgery and bioengineering. She's the founder and director of Stanford Stem Cells and Biomaterials Engineering Laboratory, and also co-director of the Stanford NIH Biotechnology Training Program. Her research seeks to develop hydrogels with unique micro and nanoscale properties to promote stem cell differentiation, tissue regeneration, and immunomodulation with a focus on musculoskeletal diseases. Her lab also harnesses biomaterials to create 3D cancer models with in vivo mimicking phenotype and drug response. Such 3D models could enable discovering novel druggable targets that would otherwise be missed using conventional 2D culture and enable high-throughput drug screening with reduced cost and time than animal models. Prior to joining Stanford, Dr. Yang received her Ph.D. in Biomedical Engineering from Johns Hopkins University and then completed a postdoctoral fellowship at MIT under Professor Robert Langer. And in recognition of her innovation, she has been recognized by numerous awards, including Fellow of American Institute for Medical and Biological Engineering, the MIT TR35 Global List honoree, National Science Foundation Career Award, Young Investigator Award from Society for Biomaterials, Biomaterials Science Lectureship Award, the Young Investigator Award from Alliance for Cancer and Gene Therapy, the Ellen Weaver Award by the Association for Women in Science, the Baxter Faculty Scholar Award, the McCormick Faculty Award, Stanford Asian American Faculty Award, and the Basil O'Connor Starter Scholar Research Award. And then it kind of ends with etc., which I thought was super funny, because I think my goal in life now is maybe to just strive for where I have to end my list of awards with etc., because there's so many more still to follow after that. That's pretty impressive. So a couple of other announcements before we get started with Dr. Yang's presentation. For any clinicians and researchers, our next virtual tumor review board for osteosarcoma, which we affectionately call TURBO, is on Wednesday, March 20th at 5 p.m. Eastern. And if you have a case to present, or if you would just like to attend, feel free to email me at christina@mibagents.org. I'll put that in the chat. And registration is now open for our Factor 2024 conference, which is going to be June 20th to 22nd in Cleveland at the newly renovated Hotel Cleveland. It is an amazing conference that brings together everyone in our community. And if you're listening to this webinar, then you definitely belong there. You will come to collaborate and leave re-energized and inspired. And I think Dr. Yang can probably attest to that because she attended last year and spoke on our preclinical models panel. We also will have a preclinical models panel this year. And some of the other topics that we'll be addressing this year are how AI will impact clinical practice and research, targeting metastasis drivers, overcoming drug resistance, key pathways in osteosarcoma treatments, local control, creating an osteosarcoma atlas, immunotherapy approaches, clinical trials, and backed by popular demand, our patient panel. So I'll put some more information in the chat about registration, but that is open now. Check it out. Walker, could you go ahead and introduce yourself?
Yeah, hi, my name's Walker, and I'm part of this year's MIB Junior Advisory Board. I got to hear a little bit about Dr. Yang's research at Factor this year, so I'm really excited to hear more today, and you can take it from there, Dr. Yang.
Okay, awesome. Well, thanks so much for having me. Let me first pull up my slide to make sure. All right. Can you all see my slides okay?
That looks great. Yep.
Fantastic. Okay. All right. So as Christina mentioned, I was very privileged to get invited to participate in the Factor conference last year. And I have attended many, many conferences throughout my career. And I must say that this is the most unique conference that I have ever been to. And I just, came out from it feeling so much more motivated to do what we do. And when I came back actually to my lab at Stanford, I actually shared a lot of anecdotes that, from the conference, and also it helped make our lab members, the researchers, to really put meaning into what we do in our everyday lab work. And I'm also sharing a picture of me and Christina at the conference as well. As you can see, we're very happy there. So I think one thing I really, I always appreciated, but I also appreciate even more after attending the Factor conference is that advances in osteosarcoma treatment really is a community effort. And, and because of that, I really appreciate this conference that bring all of these people together, right? So patients, families, patient advocates like MIB are really, the motivation for our work. And clinicians are the ones that is fighting at the front line, taking care of our patients, trying to, improve their quality of life, trying to, help with improving the survival rate. And also to develop the next generation of treatments. Traditionally, we have heard, basically the key players are usually biologists, but also more recently immunologists, right? They work together to try to figure out what's going on in the cancer. How can we figure out the magic switch that can hopefully turn things off? But I would say that more recently, people that are from the maybe not so traditional key players are also coming into this field, especially for osteosarcoma. And that includes bioengineers like myself who work on tissue engineering, but also including data scientists, because as Christina just mentioned, there are so many cool technologies, right? Like AI is becoming an important player here as well to help us interpret the large data sets we're getting from all the sequencing and try to make sense of them so that we can really utilize them to inform the drug discovery as well. So I also thought that it may be interesting to tell you a little bit about my personal background. I was born and actually grew up in Wuhan, China. And then I moved to Shanghai, did my undergrad in biomedical engineering, where actually my research back then was medical device. So again, for those of you who are still young, know that your research journey can take many interesting turns, but every phase counts and help you figure out what you like and what you're motivated. So I moved from Shanghai to US afterwards to pursue my PhD, also in biomedical engineering at Johns Hopkins University. But here I actually get to do research rotations, which allow me to explore fields that I never had an opportunity to be exposed to during my undergrad. And that's actually what made me actually switch from medical device to actually a field called tissue engineering. And in particular, when I was there, I worked mostly on bone and cartilage tissue engineering. And afterwards, I moved further north up to Boston and did my postdoc. I spent 3 years at MIT, and that's where I actually worked still on biomaterials as a tool, but in this case, a very different application. I was working on non-viral gene delivery. And since 2009, I have moved to Stanford University. So in this case, I moved to the West. And I have been very happy here for the last 14 years running my lab, focusing on developing stem cell and biomaterials therapies for various applications. This is actually my lab after we moved into our more recent lab building. You can see we're a pretty fun group. And this is one of our Chinese New Year celebration with all the red envelopes. We also had dim sums this year. These are some of our more recent lab retreat photos where we actually go to a local a place that is usually within a half-day drive. This is at a farm, and this is actually at Monterey Beach, where we really just talked about what are the opportunities, what are the challenges, and how can we contribute and synergize as a very interdisciplinary group. So the title of my talk today is Tissue Engineering Strategies for Elucidating Osteosarcoma Biology and Informed Drug Discovery. I understand our audience comes from all different stages and different backgrounds, so I thought it might be worthwhile to take a minute to just explain to those of you who are not familiar with this term, what is tissue engineering, right? So this term was actually first coined back in 1988 at a very small symposium. And the idea was to apply principles and methods of, as you can see here, engineering and life sciences. So really from the very beginning, this field requires the collaboration and crosstalk between engineers and biologists. And there are two goals for this. One is more for mechanistic part, is how can we better understand how tissue develop, whether it's healthy or unhealthy. And the other goal is to actually for translational. Right? How can we then use the knowledge we gained from these tools to develop basically tissue substitutes to help those people who have lost tissues and organs due to disease or trauma? And this is also a landmark paper, actually from almost 30 years ago, published by Bob Langer and Joseph Vercanti from MIT and Harvard, respectively. So this is also an interesting slide I like to share. I'm actually teaching a course called Tissue Engineering at Stanford. And this is a slide I always like to show to think about history part. So Dr. Langer is actually was trained as a chemical engineer, and his specialty is actually making biodegradable polymers for drug delivery applications. And Dr. Vercanti is actually a liver transplant surgeon. And one of the challenges that he has in his everyday, clinic is just like there's never enough organ donors to help with organ transplant. So one of the issues that he is dealing with is how can we get more tissue available so that we can help the patients who are just on the wait list, right? And they happen to be good personal friends. So they were talking to each other on vacation. And then Dr. Langer said, oh, we have these polymers. How about we make them into a three-dimensional structure where we can put cells in, right? And then use them to grow the tissues that you might need. So that was kind of the initial, I would say, the birth of the concept is to take cells from different tissues and then to grow them actually in a biodegradable polymer scaffolds that is three-dimensional. And then you can, if you give them the right signals and conditions with the right time, the goal is that then we can actually have a piece of bone, cartilage, liver, whatever people need that transplant surgeons can use to address this, limited, supply problem. And because of that, when you think about general concept, when we do research in tissue engineering, there are a couple of key components that we typically think about. One is just cells, right? They are the ones who make the new tissues. But cells themselves are oftentimes not enough because they can easily die. They really need support. So that's where the scaffold or the biomaterials comes in. And the idea here is that we can make different polymers into a unique three-dimensional structures that can then guide the cell by modulating, by learning from what we already know from developmental biology. So eventually we can have some tissues that we can put into animals and eventually go into the patients to help with restoring the tissues. And this is really the fundamental kind of, I would say, the concept that drives a lot of our research is that we try to, really understand, what cells how do cells make decisions depends on what they're talking to, right? This could include soluble factors they may be exposed to during the development or disease. And this also involves usually the communications among multiple cell types. And a very important part of the player here is actually extracellular. To keep it simple, but this is really important because the ECM is actually what the cells are residing in as their use is. They actually can then become either regeneration. So we apply a lot of this knowledge that we gain for either, regenerating tissues, for disease models, especially cancer, but also for studying how can we regenerate tissue better for patients that are actually aged, because usually for younger people, they can regenerate on their own. It's really the aged population where a lot of this is very, very difficult. So with that intro, I hope that gives you a background of, where I came from and how this field could potentially be helpful. Is that in my lab over the years, we have developed many different biomaterials, and I'm just showing some examples here. And then we use them traditionally for regenerative tissues and also for drug delivery applications. But then, tools, one opportunity that we identified is that this can also be really useful for studying cancer by mimicking the cancer microenvironment. And also this could be really useful for immunomodulation as well in the context of both regenerative medicine and cancer disease modeling. And I have students in my lab work on both directions. So today, in the time that we have, I'm just going to tell you one area, which is applying the tissue engineering principles for studying bone cancer, in particular osteosarcoma. So osteosarcoma arises from bone tissue, which is actually a very unique niche for cancer growth. So here we're looking at a cross-section of a long bone with the bone marrow in the center, right, surrounding by the very dense bone matrix. And when you look at the bone niche, it actually has very unique mechanical environment. As we know, bone is actually much harder tissue compared to, muscle or fat, right? So that mechanical signals also matters when it comes to tumor, biology and drug response. But as I mentioned earlier, In the tumor niche, in addition to the tumor cells, there are also other cells such as osteoblasts, osteoclasts, and other immune cells. And very importantly, all of these cells don't just exist in the, in open space. They actually reside in this very dense extracellular matrix. And in the context of bone, actually, a majority of the bone ECM is actually made of inorganic minerals. And that's what makes bone very hard, much harder than the rest of our tissues, and also more brittle. And it also has about 30% of collagen. So when it comes to studying cancer, generally the current standard of art of experimental models using either just growing the cells in a 2D tissue culture plastic plate, which is what we refer to as 2D culture, or using basically animal models. As we know, 2D culture is really convenient, and they were actually the standard, and they are still actually mostly the standard these days in the biotech industry when it comes to high throughput drug screening because of its low cost and ability to support, screening hundreds, thousands, or even, millions of combinations. But the challenge with the 2D culture is that, because it doesn't have the three-dimensional structure, there's no ECM here, there's very, tunability. While they're very easy to do, a lot of the drug hits identified from this approach basically fail as they move down the pipeline. And that's why a lot of the drugs actually, don't work as we move, through the letters. On the other hand, animal models are much more physiologically relevant, right? They have all the different cell types, the matrix there. But the challenge with the animal models is that they're just very, very low throughput. It takes months for the tumor to grow before we can even do any treatment. And it's also very expensive. So this is where the opportunity arises for tissue engineering, is that we could actually learn from a lot of the tools we have developed originally for actually regenerating tissues, in this case bone, but apply them to actually study how cancer, especially cancer that arise from bone, actually, develop in the bone niche. So when I had this idea, a while back, I actually thought about, okay, this is not something that we can do completely on our own. We need to learn and collaborate with other experts in their own respective disciplines. And this is when I actually just reached out to Dr. Alejandro Sweet-Cordero, who is also in the Bay Area. We're at Stanford, they're at UCSF, so we are actually not too far from each other. I actually didn't know him personally at all. I just sent him a cold email and said, look, we do tissue engineering, we do biomaterials, we have started to look into osteosarcoma, but we would really love to learn from you, more about the biology. But also, I want to ask him the question, do you think this is even useful, from your perspective? Because he's a clinician, he's also a cancer biologist, and also, his lab does a lot of sequencing as well as biostatistician statistical analysis. So he actually responded to me, I think, within 20 minutes, even though we never met and we both had busy schedule, saying that absolutely, we need tools like this. And that's how our collaboration started, from a few years ago. And our lab has since, started to collaborate very closely. So previously, when people, people actually have thought about, can we move from 2D to 3D for osteosarcoma? And majority of the research before was using actually just by putting the tumor into a centrifuge tube and centrifuge them to be a spheroid. So this is just a tumor aggregate. Or people use some hydrogels or scaffolds to try to grow them in 3D. The limitation, though, of this approach is that, number one, majority of the research used very few actually cell lines that are established, but they have been cultured just in 2D for decades. So they no longer really capture the patient, tumor biology. And as we all know, one of the key challenges of osteosarcoma is actually heterogeneity, right? So how can we really use cells that are more relevant to study this is really important. And also another limitation of the previous work is that outcome analysis were very, very preliminary. And there's also no really in vivo validation when it comes to, drug effects. So this really drives the goal of our work when we started this, is that we want to actually really engineer a three-dimensional scaffold that mimics the bone ECM environment. We want to focus on patient-derived cell lines that represent better their biology, but also heterogeneity, and then integrate that with, high-dimensional sequencing technology, and also, to validate whatever 3D specific drugs that we can find. We want to also validate that using mouse models to see, if this actually does translate. So when we started this work, the first thing we did is actually to just look at the public available data to compare osteosarcoma versus all the other different type of solid tumors to see what are some of the signaling pathways that are enriched in osteosarcoma. And to our I guess in this case we were very happy to see is that if you look at the signaling that are really highly ranked, they're actually collagen, bone minerals, right? That these are actually important players in the bone extracellular matrix. So this really again support our initial hypothesis is that bone ECM probably plays a very important role in osteosarcoma biology. And this really is very poorly studied before, so it's an important opportunity for us. And the questions that we were asking here is, how does bone ECM impact osteosarcoma biology by looking at signaling, but also what are the implications here that we can learn from these 3D models that can help us discover drugs that are more useful. So this work was actually initiated by a very talented recent PhD alumni from my lab, Dr. Eva Gonzalez-Diaz. And when we started this project, she looked at basically what is the right materials that we should choose for making these 3D scaffolds. So again, we learn from the biology. So in the bone niche, we know that it's about 2/3 minerals and about 1/3 of them is collagen. So mineral and collagen are really the bread and butter here. And then we came back to our lab and we have a lot of different biomaterials tools, as I showed you earlier, right, that we have developed before for actually bone tissue engineering. And we say, okay, awesome, we can learn from those and use our existing tools. In this case, we chose a biomaterials called gelatin-based microribbons. And then we can mix them with another kind of signal called hydroxyapatite, which basically mimics the mineral part of the bone niche. And we can mix these materials together and then also mix them with the osteosarcoma cells of choice of different patients and then grow them in three-dimensional space. Now, with this three-dimensional model, we can then look at the effect of transitioning from 2D to 3D, but also to look at effects of compositions of with and without mineral and compare that to in vivo mouse models when it comes to different tumor biology and drug response. So as I mentioned, the 2D model are typically not very good at predicting drug response. And this is actually very well illustrated in this paper published actually by a group from Stanford, where they're looking at a correlation of, basically drug response and biology from different models. So here you can see the yellow line is the 2D model, and the red line is the patient tumor samples. And the blue line is 3D culture. You can see the 3D much more closely mimics the tumor from the patient, where the 2D is much, much separate, right? And the mouse model is actually somewhere in between. And here is a list of different osteosarcoma cell lines that we have been working with that have been provided to us from our collaborators with Claro's lab. And they have spent actually over a decade just to generate these patient-derived models, but also have very, very deep characterization using various sequencing technology. So this really gives us a very strong starting point when we start this project. The first question we ask is that what is the effect of this three-dimensional culture and also the incorporation of mineral into the 3D culture on osteosarcoma tumor proliferation, right? Because so here, what we're looking at first is the structure. So on the left, you can see that this is the morphology of our microribbon scaffold. You can imagine them to be kind of like fettuccinis, right? So these fettuccinis can actually cross-link together. You can imagine the cells are like the meatballs, and you can bake a lasagna together, right? So here we can basically also make our cell-encapsulated microribbon scaffolds. And when we incorporate minerals, you can actually observe the deposition of these mineral particles on the surface of this fettuccine gelatin hydrogels as well. So very importantly, we first verify that cells are viable. So here we're using a live death staining. So you can see the cells are very happy. They can grow in all of this culture, but the speed of their proliferation is very different. So here you can see that in 2D, these tumor cells just grow crazy. They really become very confluent very quickly, which is very well known because they're very aggressive. But if you look at the in vivo tumor growth, though, which is illustrated here by the orange line, the tumor grows, much slower, right? It takes actually usually a few months for the tumor to become a mass in vivo, which is why the animal models are also a painful one to work with. But what we actually see is that when you have a very artificially fast-growing tumor in 2D culture, that actually artificially changed the drug response. So that's partly why 2D model usually doesn't really show much drug resistance. So here we see that at least, from a proliferation level, we see that 3D culture definitely, better mimics the tumor proliferation in vivo. And also we saw that when we incorporated the mineral cues, which is present in the bone, it actually further slowed down the tumor proliferation that is closer to in vivo as well. Next, we asked the question about, what is the effect of this culture on actually drug resistance, right? Christina mentioned that's one of the direction of this year's conference, and it's a big challenge for many cancer, including osteosarcoma. So here you can see that when we look at a drug response curve of using different culture models, you can see that, at the same concentration, the cells die a lot more in the 2D model. They show really, very sensitive to the drugs. But when we move them to the 3D model, and also when we incorporate the bone ECM, especially mineral cues, these cells become a lot more resistant, which is actually closer to what we typically see with mouse models. So on the other side, you can see that here we're also looking from the gene expression level to look at how does different culture model change the drug resistant, protein expression. And here we can see that very consistently here we see the three-dimensional culture and also incorporation of the bone niche cues really significantly upregulated this drug resistance protein expression. So this further highlights the importance of using the 3D model to study drug resistance as well. And then another signaling pathway that we were very interested to look at the beginning is actually a pathway related to IGF. And the reason is because IGF-1R signaling has been demonstrated in multiple cancer types that they are really the key drivers for tumor progression, but also drug resistance. In fact, another tissue engineering group from Rice University has actually compared the 2D and 3D model before for Ewing sarcoma. And they show that when you actually culture these Ewing sarcoma tumors in 3D and also in vivo, you see definitely a significant upregulation and activation of the signaling pathway. And in contrast, a lot of this is actually largely, lost in 2D culture. So we said, okay, let's study this in osteosarcoma then, which is the disease we are interested in here. So here again, we are looking at different assays. So on the left, you're looking at the quantitative value of the protein level in 2D or 3D model, as well as using in vivo subcutaneous tumor as a positive control. So here we can see that, again, similar to what was previously reported with Ewing sarcoma. Here in the osteosarcoma case, we also see that the three-dimensional culture was really important for actually retention of this signaling. On the right, you can actually see immunostaining. So this is actually allowing us to visualize the distribution of the signaling expression in addition to just the quantity. The blue signal here stands for the cell nucleus, and then this brighter signal is staining the protein expression by the cells. So you can clearly see a very consistent trend that, again, 2D culture, this signaling is largely lost, and 3D culture is more. But also one thing I hope you could appreciate here is that when we have the minerals, this signaling activation is even further enhanced, right? And this is actually one of the question another experiment we did here is that when we actually deactivate this signal of IGF-1R using an inhibitor, you can see that now actually the drug resistance is actually largely abolished. So again, suggesting there's a close correlation of this signaling activation with the drug resistance. So another question we also asked is that we have observed that mineral actually is important, right? But what about the dosage? Is there optimal dosage of mineral cues that we should incorporate here? So this is actually where we compare different dosage of minerals, and we identified that actually once we reached the intermediate concentration, then it kind of was sufficient already. So that's how we then chose that concentration for further studies. The next question we then ask is that, how about, can our model actually predict the osteosarcoma signaling in vivo? So one thing we noticed from our in vitro model is that the expression level of this target is actually higher in our gel HA model versus the one without the hydroxyapatite, right? And it's even higher than our in vivo positive control, which is the subcutaneous model. And we thought about it. Well, subcutaneous model, people use it a lot because it's easier to do. You just inject into the subcutaneous space, right? And you can do multiple tumor per mouse. So it also increases the throughput a little bit more for animal screening. But if you think about subcutaneous space, there's no bone matrix there. But in our in vitro model, the gel model actually has no mineral, and the gel HA has mineral, like the bone, right? So from this in vitro data, it actually gave us a hypothesis that actually, if you go in vivo, we would expect there will be a difference in this signaling if it's subcutaneous tumor versus if it's in the bone-ish, which is what we call orthotopic tumor. The reason people don't use orthotopic as often is because it takes much longer to grow, and it's also quite difficult to work with. It's a much more tricky surgery because you inject actually into the bone niche. They grow much slower than the subQ tumor, so we have to wait longer. And you can only do one tumor per mouse rather than subQ, you can do multiple. So that's the reason people usually use subQ, because of the throughput and convenience. But our data from the 3D model here really inform us that, you know what, even though it's inconvenient, but it may be important for the biology. So we then went back to the in vivo and characterized the signaling there. So here you can see that we're testing with 2 different patient-derived osteosarcoma cell lines. And then we compared subQ tumor versus the ortho tumor, which is in the bone niche. You can see that for both patients, they demonstrate very consistent trend, is that when the tumor is actually residing in the bone niche, this signaling is significantly upregulated. And this corresponds very beautifully with our in vitro model. So this is actually very nice. It shows us the power of actually using a simpler and much cheaper and faster 3D model to help us actually inform what might happen in vivo, to really save the time, but also help us narrow down the right experiment model to use. Why is this important? Well, if you remember the data I just showed you, right? We showed that in the subQ model, the signaling is less, which also correlates with less resistance. But when we go in vivo, now we actually see that indeed, when we look at tumor viability with the same treatment, the tumor shows much higher drug resistance in the orthotopic model as well. And this trend is consistent across patients. Here I'm showing 2, but we have done more than 2 as well. So together, this is very important because it shows us the importance of using 3D culture for in vitro with the right bone ECM. It also shows us the importance of using actually orthotopic model for in vivo as well when it comes to drug validation. So those are some of the examples I showed, but I also want to show you some of the other assays that we could do is to integrate our 3D model with some of the high throughput, either protein-based or gene sequencing-based assay to help us understand the effects of culture model on their, signaling pathway. So here, in particular, we're looking at the RTK phosphorylation of multiple RTK, targets. And we're just generating a heat map to illustrate their relative expression level. So one thing that is very clear, again, across different patients, is that the 3D model correlates much better with the orthotopic tumor, whereas the 2D model really shows a very different trend. And this is also true at the gene expression level when we performed high dimensional RNA sequencing. So here, again, we're looking at a cluster analysis. So you're looking at which kind of culture models shows overall the similar pattern of gene expression. We're not looking at just one gene here, but we're looking at thousands of genes, right? So again, here across multiple patients, we see that the tumor from the patient, which is the purple one, cluster much closer with our 3D model. And in multiple cases, they cluster the best with the gel HA model, which again, give us further confidence of using this for our future studies. And as I mentioned, data scientists are very important here. So working with also a very talented data scientist from Alejandro's lab, we also performed additional pathway analysis to compare, how much signal are overlapping across different models. So here, tissue is the group representing the patient tumor. And this is 3-dimensional gelatin versus 3-dimensional gelatin HA. And you can see that, again, this shows us that the three-dimensional gel HA model has the most overlapping signaling pathway as the patient tumor as well. And this approach also allows to identify some of the top signaling pathways that are enriched in 3D and patient tissue compared to 2D. So the idea here that we're pursuing is that using this 3D model and also by integrating it with RNA sequencing analysis, we could actually target, drug candidates that are upregulated in 3D specifically, as well as in patient tumors that are actually lost in 2D. So this would allow us to actually, potentially identify drug candidates that would be missed out if we're using 2D culture models. So here is just demonstrate one example that, using this approach, we were able to actually identify a potential drug candidate, alpelisib, which we then performed the drug screening in vitro using various models of 2D and 3D. And we actually showed that in this case, the 3D gel HA model actually showed the best drug sensitivity. So we then went in vivo to compare and to validate, which actually took. So one thing I want to put in perspective, this experiment takes a few weeks. This experiment takes a few months because, we have to wait for the tumor to respond, and we have to do the dosage and to wait for much, much longer. So, but what is very nice here is that we were able to use this 3D model to actually help us narrow down the candidates that also get validated in vivo to show great, efficacy for inhibiting the tumor growth. So currently, we're actually applying this technology for studying multiple patient, osteosarcoma tumor cell lines to understand the heterogeneity and also to identify the drug candidates that target this kind of heterogeneity. So just to summarize, here, what I was hoping to share with you is that, tissue engineering can be a very useful tool for studying osteosarcoma. And in particular, what we have found is that three-dimensional culture as well as bone mineral cues actually are very critical for modulating osteosarcoma signaling as well as their drug resistance. And also when it comes to in vivo, we actually found that even though it's very painful and slow to do orthotopic model, it actually is worthwhile because they actually have much better physiological relevance. We also demonstrated that RNA-seq is very useful for integrating it with this 3D model to help us inform drug discovery that could otherwise be missed out in 2D. So apparently this is only, I think we're still at an early phase of, dedicating our expertise and experiences for osteosarcoma. We're actually asking various questions, thinking about how tissue engineering tools can be helpful for future directions of osteosarcoma. I think one thing I already mentioned is this high genomic complexity and heterogeneity. So I have a very talented postdoc in my lab who is actually working very hard on optimizing our model for supporting, multiple cell lines, and then to also do drug screening that can target this kind of heterogeneity. It's definitely not an easy task, I can tell you. That is very challenging. We're still in the middle of it. Another question that we're asking is that, well, because osteosarcoma is known to be very easy to develop drug resistance, we expect that combination therapy is probably necessary, even with the target therapy, right? So this is one thing that we're also hoping to pursue, is to use the 3D model to also test, leading candidates that identify from target therapy with some of the more traditional chemotherapy to look at, what combinations actually could also be very promising. And we could also use this to study drug resistance because, as I showed you earlier, these cancer cells grow actually artificially much faster in 2D, and they actually do not retain the drug resistance, mechanism. So we could actually use this 3D model and integrate it with various other tools to identify, potentially what subpopulations of the tumors within osteosarcoma are responsible for such resistance. What are their molecular signature so that we can actually develop better therapies there. And I also have another very talented PhD student in my lab from Bioengineering who is now trying to incorporate actually either additional niche cells from the bone niche, such as osteoblast, as well as tumor-associated macrophages into our model. Now, one thing I do want to point out is that there's a balance here, right? The more cell types we put in, the more physiologically relevant it is. But the challenge is that it will decrease the throughput of the drug screening. So it depends on the question we ask. We also want to balance that complexity. And finally, we know that like many cancer, the main challenge and the most difficult part of osteosarcoma is actually metastasis. And this is where my lab actually has developed a different tissue engineering platform that is called spatially patent, cancer metastasis model that we have demonstrated great efficacy in looking at breast cancer bone metastasis as a proof of concept. And we think that it's a model that can also potentially be very useful for studying osteosarcoma metastasis in the future. So just to give you a little bit of flavor of this other platform, so here we're trying to mimic the breast cancer invasion from the bone marrow into the surrounding long bone tissue. So to do that, we actually use our tissue engineering approach to make a tissue engineered bone in the lab. And then in the center, we can use another biomaterial to mimic the bone marrow where we introduce the cancer cells. So here the cancer cells are labeled green and the surrounding bone cells are labeled red. So then we can use this model to actually look at over time, how do the cancer cell actually invade from the bone marrow space into the surrounding bone tissue. So here we actually could make the outside, the ring, to be different tissue types using tissue engineering approach that is either bone or cartilage or muscle. So for breast cancer, for example, and also prostate cancer, they're known to have very, bone-specific invasion. The chance of invading to bone is much greater than other tissues. So that's actually the first thing we demonstrated with this model is that our model actually predicts this tissue specificity in cancer invasion, is that only when the cancer cells are surrounded by tissue engineered bone, they show a lot of invasion. But when we switch the tissues outside to be cartilage or muscle, which are actually made from stem cells and biomaterials, the cancer actually do not invade. So this is very encouraging. And also we know that some cancers are actually much more likely to invade or invade much faster, right? But how do you predict that? It's actually very difficult. So here we actually use very well-characterized cell lines that are either more aggressive or less aggressive across two different cancer cell types. And our data showed that, again, in this model, the more aggressive cancer actually invade much faster into the surrounding bone. So this actually also suggests that this could be very useful even for a diagnostic tool, because the clinicians could potentially then know better, okay, this patient's tumor is actually not going to invade as faster. So maybe we could be using, therapies that are not so aggressive, right? So this is a great way to help us understand what might happen down the road. And also very importantly, one of the reasons that cancer metastasis actually in this case is very lethal is because when they invade into the bone, they actually destroy the local homeostasis of the tissue and they start to destroy the bone. So in this case, we actually use micro CT to monitor the tissue engineered bone and then look at the bone density over time. So here you can see at the beginning, you have this circular bone outside. But then over time, when you introduce the cancer cells, over time, the bone actually gets destroyed just as if it's happening in vivo as well. So again, this shows the model can predict or can mimic the cancer-induced dysregulation of the bone remodeling. And finally, we also demonstrated using a drug that has demonstrated efficacy using animal models before called PTH, parathyroid thyroid hormone. And we demonstrated again, using our model, we can actually mimic the benefit of the PTH in reducing this cancer induced basically invasion as well as destruction of the bone. So this is a model that right now we're actually integrating it with also barcoding as well as single cell sequencing technology, because the question we're asking is that now we have a tissue engineer model that can mimic the cancer metastasis, right? In this case, into the bone. We could actually use this to isolate the cells that are more invasive, that actually kind of taking a lead, right, in invading and infiltrating into the normal bone tissue. And we can use single cell sequencing to understand, what are the molecular signature that actually define the highly invasive cells. And these are some examples showing that indeed the cancers that are much more invasive, they demonstrate upregulated gene expression pattern related to invasiveness, stemness, as well as ECM modeling. So again, further highlighting the physiological relevance of this model. So going forward, I think one of the opportunity that we think could be very interesting to pursue in the future is to use this experimental model for actually studying osteosarcoma metastasis. In the case of osteosarcoma, though, their metastasis, one of the major sites is actually lung tissue. So, one potential idea that we're thinking about is to actually, engineer lung tissues in the outside and then introduce the osteosarcoma that mimics the bone niche in the inside, and then use that to actually study osteosarcoma, tissue-specific metastasis. So with that, I would like to stop here and to acknowledge my lab. And also in particular, most of the work that I mentioned today were done by Ebba and in close collaboration with Sweet Caldero's lab at UCSF. As I mentioned, this is ongoing work. We were very grateful that NIH NCI actually has funded this idea of integrating tissue engineered 3D model with basically sequencing tools to better understand osteosarcoma biology, and also we're working very hard to discover drugs that can target heterogeneity as well. So with that, I'm going to stop here, and I'll let Christina and Walker moderate the questions.
Thank you so much, Dr. Yang. Amazing presentation, great explanation, and it makes me want to start, like my fourth career as like a bioengineer.
Well, we do have probably, younger people that are either in the audience now or maybe looking at this webinar in the future. And I hope that this would, help them also, get inspired to get into this field, because it's a very difficult problem. And I think we need the more people that can join us to study this, the better.
Yes. Well, we have lots of questions. So Walker, why don't you go ahead and start tackling those, and we'll try to get through them all.
Yeah. So the first question that came in says, interesting work adding the HA. Did you also investigate downstream signaling of IGF-1R, such as PI3K, AKT, or mTOR in the scaffolds? And then along with that question, they also ask, were IGF-1R or mTOR inhibitors also tested in the in vitro system?
Well, that's a great question. We actually did look into some of them, but not all of them yet. And that's something that we definitely would like to do with the ongoing work. And as I mentioned, one of the things that we're also trying to understand is the heterogeneity here across the patients. So that's actually another thing, is just, we have to also scale this to test with multiple patients to see if we see consistent trend. So that's something that we're working on very actively right now, and those are great suggestions.
Gotcha. Another question that came in asked, how much time did it take for the orthopedic model to develop in mice? Were there any lung mets? And if yes, after how many days?
That's also a great question. I can tell you that varies a lot. And it actually is because of the patient tumor varies a lot. So across the 8 tumor cell lines that we're working like right now, I would say the fast one, the tumor, probably will take about maybe 30 to 40 days to develop to a certain size before we can start treatment. But some of them are growing really slow, and it can take 200 days for the tumor to form. So this is just a very wide range that actually represent the true biology in patients, right? So this is also something we're trying to study, is that could our 3D model also better mimic this kind of variability when it comes to the osteosarcoma proliferation, because I do think it ties very closely with a lot of the drug resistance as well as their drug response.
Yeah, that makes sense. Another question that came in asked, did you look at how this can be incorporated to assess Borg, the primary tumor in matched metastases?
Oh, can you repeat that again?
It says, did you look at how this can be incorporated to assess Borg, the primary tumor in matched metastases?
I think it might maybe mean both.
Was it
That's both.
both? Oh, sorry. I was like, I don't know.
Okay, so right now we are focusing on looking at osteosarcoma as a primary tumor and looking at the drug response. But as I mentioned in kind of towards the end of my talk, I do think that in terms of the need, I feel like the metastatic tumor is actually more difficult, and is where will probably be even greater impact. So that's why I introduced this other model that we have developed, not for osteosarcoma, right? It was actually for looking at breast cancer bone metastasis. But I do think that the concept there is easily adapted, can be easily adapted for studying osteosarcoma. And we haven't yet worked very actively on that. Just we have our lab of maybe 12 people. So, but it's a project that I have personally, I'm very, very interested in pursuing. Probably we just need another talented postdoc and PhD student to work on it.
Gotcha. Another question that came in says, in your sub-Q and orthotopic models used, what is the number of tumor cells injected and tumor latency?
So that I don't remember some of the details there, but it's in our published paper in Advanced Healthcare Materials. Some of the method details you can whoever asked the question, please feel free to look it up in our paper.
Gotcha. Another one says, magnificent presentation. Thank you. How do you control the psychochemical environment throughout the 3D construct? What about the non-collagenous proteins that are essential in the bone microenvironment? How does the self-assembly influence the pore size, surface area, etc.?
Well, those are all excellent questions. Those are a lot of questions too. I guess maybe take a step back. I think one of the other ongoing project in my lab is to also ask the question on what is really important in that three-dimensional environment, right? So I mentioned using the gelatin microribbon system plus the hydroxyapatite, which is unique and invented by my group. But we're thinking, there are also other biomaterials out there that people have used for bone tissue engineering that could be used for osteosarcoma, right? And the study is kind of very sporadic. Each group used just their own material, and there's no really comparison. So one of my PhD student is actually doing a comparative study right now by looking at multiple different biomaterials that people have used for bone tissue engineering, including some of them are just very easy and commercially available. Some of them may need a little more biomaterials fabrication. But the question we're asking there is actually related to the question, is that we're asking, what is the really important thing? Is it just the three-dimensionality? Is it actually the porosity, right? So we compare the material that are either nanoporous or microporous. We also looked at, what about biochemical cues? Is collagen really essential? What if it's a three-dimensional gel that doesn't have collagen, right? So we compare different kind of signal in the 3D biomaterials, trying to understand part of that. But I would say that the goal here is not to decouple the niche cues, because many of them are also confounded. Like you change one thing, you might be changing the other. But the main goal or the main motivation of that project is that we're trying to help the community understand what are the, multiple biomaterials that people may be able to use, depends on what they can do, what is accessible, so that they can use those 3D models for their osteosarcoma work in their own lab as well.
Gotcha. Another question that's come in asks, what mice model can you suggest is best for osteosarcoma, Balb/C, nude, or C57BL/6?
Well, in our case, we are using immunocompromised mice because, we're putting patient tumor into them. So that's that basically limit us to NSG is the main one we're using for immunocompromised mouse model.
Gotcha. Another question asks, have you tried a comparative analysis to see how 2 or 3 of the osteo models differ?
Can you repeat that question again? I'm not sure I fully understand it. Yeah.
It says, have you tried a comparative analysis to see how 2 or 3 of the OS models differ?
In terms of what does the 2 or 3 models mean? Does that mean the different patient cell lines, or?
I guess so. They didn't
So
really
we do,
specify.
yeah. So again, that's actually one part of our ongoing grant is to integrate a model to study the heterogeneity. I would say that's also where we're trying to understand, one of the ongoing project also is that we're actually focusing on the drugs that are currently in clinical trials for osteosarcoma or in preclinical models. Because one of the things we're trying to understand is that we know that, these drugs don't work for everybody. but people don't understand the correlation. So one thing we're using our 3D model is that we can use our 3D model to grow, multiple patient cell lines, right? And test their responses to these drugs. So for some of the drug, we see that, only 2 cell lines work. Most of them don't respond. Other ones, we may see more responses, right? And we also have the sequencing data of these cell lines. So one of the power of using this 3D model is that we can start to gradually pieces together the puzzle. Is that, what kind of either genomic feature or maybe epigenetic feature that may correlate the cell lines that respond to certain drugs, but not to others. So this may help us to, again, inform, like the drug kind of precision medicine going forward as well. But it is a very challenging problem to work with, again, because the heterogeneity is high. So we're trying to also figure out, how many do we have to test to be able to actually, find a pattern. So it is a very challenging problem, but we're working hard on it.
Sounds good. Another question that's come in says, how can you study the role of immune cells in the bone environment with your 3D model? They have a significant important role in development of osteosarcoma. How can you correlate with your 3D model?
I love this question. As I mentioned earlier, I actually have a PhD student and her thesis is on this. And in particular, right now, we're focusing on tumor-associated macrophages because within the osteosarcoma, so there's actually a lot of clinical sequencing data using patient tumors, and they show that actually, tumor-associated macrophage is actually accounts for a large population of the immune cells within the tumor microenvironment. And one of the questions we're asking here is actually how does the cell crosstalk influence the tumor-associated macrophage polarization? Because this macrophage, they're actually not committed. Depending on what they see, they can become either this way or that way. So they can become either more of a tumor-supporting immune cells, or if you actually can polarize them, can control them, they can actually help with tumor killing as well. So that's actually one thing. The general concept we're pursuing here is to add these immune cells into our 3D model. Obviously, we have to do some engineering optimization here to support that. But we want to first demonstrate the model can mimic the tumor supporting versus the tumor killing, subtypes, and then use this model to test what if we can use drugs to modulate that? Could this be another alternative? So instead of directly killing the cancer, we could actually engage the helpers to kill the cancer, right? So this is another approach that involves a lot of collaboration with immunologists as well. But it's a great question, and we're definitely thinking along the same way.
Oops, I think maybe Walker might have been frozen there. But we have a couple more that I think we can try to get to. So let's see, one of the questions was you used PTH in one of the slides. Have you tried to see how 1,2,5-hydroxy vitamin D would do?
Oh, we haven't. But that particular model was not osteosarcoma as well. That particular model was actually for breast cancer bone metastasis. We chose that particular drug because it was demonstrated with efficacy using mouse models already from our collaborators lab. So we want to show that, okay, at least can our 3D model recapitulate what is already working in vivo. But absolutely, I mean, the concept here is that if there are any drugs with demonstrated efficacy either in patients or in animals, right, those are the ones that we want to test with our in vivo, with our 3D model to show that there is a good predicting power. And then with that, then we can use it to screen, potential new drug candidates. But one thing I do also want to point out is that I don't think any 3D model in vitro can completely recapitulate in vivo either. So I want to set expectation reasonable, is that in vivo, there are just a lot of other things going on, right? But what we're trying to do here is to have something that is filling the gap between the 2D model, which is the current standard, versus the animal model. So compared to the 2D model, we have definitely much more physiological relevance, but compared to the animal model, it is much more scalable. We can do drug screening much faster. I also want to point out that 2D model has its value as well. It's not like 2D model is completely useless. There are certain signaling that 2D is actually very good at retaining. And in that case, it's actually better to use the 2D model because it's cheaper, it's faster, right? So I think, again, every tool should be used for addressing the right questions.
Another question coming your way, a couple more. Do physical force factors, shear force, for example, acting on the 3D models, have any influence on these findings?
That's a very good question. We haven't looked into that, but I can tell you again from my background in tissue engineering, there are bioreactor design where people can incorporate them with this kind of tissue engineering 3D models. So you can actually apply whatever mechanical stimulation you want, shear force or mechanical loading, etc. In the case of actually tissue regeneration, for example, people have demonstrated that if you're putting stem cells in a three-dimensional scaffold, and if you apply cyclic loading, for example, it actually can enhance their tissue formation. So again, osteosarcoma is a tumor that is also subject to mechanical loading as well. And that's definitely a valid question. It's not something we have looked into right now, though. Maybe someone else who have the bandwidth, who is a mechanical engineer, can definitely take on that.
And we're already at time, but you can see there was a lot of interest in your presentation. I wanted to just ask a couple last questions before we have to go about just like, again, comparing to the 2D models in terms of like how much tissue is needed to be able to create, like, is there any advantage? Is it easier at all? Because I know sometimes it's, there's not like 100% success rate, right, in creating a 2D model in the dish. And I'm wondering if it's easier to create the ECM model. And like, do you need more or less tissue? Is it the same? Is there an advantage?
That actually is not, well, I guess if I understood the question correct, it's asking that if a patient had a surgery and, had some tumor, right? Like, how could that be derived into a cell line or xenograft model? That actually is what my collaborators lab has spent the last decade to do, is that they would get patient tumors and then they would first engraft that into the mouse. But one thing I do know is that the variability is very high there. Some of the tumors just don't engraft, and you can never derive that into a xenograft, right? So out of probably many tumor samples they have worked with, I think now they have a few dozen of tumor xenograft they were able to generate, but it took years of work to get those tumor generated. And once you generate them in the mouse, then from there, they can be derived into cell lines that can be cultured in 2D, or we can then grow them in 3D.
Yeah, so I guess my question would be comparing like going from PDX to a 2D or going from PDX to your model. Is there any advantage in terms of time and even just like the tissue?
I think initially I would say it actually is easier for 2D, because for 2D, you just put cells in onto two-dimensional plate. And you can use like 96 well plate or smaller plates to do the screening. In 3D though, it's a three-dimensional space, right? So we actually need a lot more cells to make our 3D models. So one thing we are doing though is to miniaturize our 3D samples, so that we can, with the same number of cells, we can actually do more combinations and more screening, because you have to do replicates, you have to do different dosage and different analysis. But from the number of cells for 3D study, we actually have to expand the cells first, so that we have more cells to work with. But this actually resembles the in vivo tissue better as well.
Right. So much innovation. Thank you so much.
My pleasure. Thanks for having me.
Yeah, really appreciate your joining us today and making it better for bone cancer patients. More information on this and all OsteoBites can be found on our website at mibagents.org, on our YouTube channel, and at your favorite podcast place. And next Thursday, we're going to be joined by Dr. Jennifer Mack. She is Associate Chief, Division of Population Sciences at Harvard Medical School, and she will be joining us on OsteoBites to discuss patient-powered research and best practices for approaching patients to participate in research and clinical trials. You can find our OsteoBites lineup for the next few months on our website, and if you have any ideas for future topics that you'd like to hear about, please share them with us at events@mibagents.org. For sure, Dr. Yang, we're going to have you back after your lab has had some more time to work on all those amazing projects that you guys are working on, to kind of hear about the progress. Thank you so much for joining us today, and thanks to all of you for taking an hour and spending it with us today. We hope to see you back here on OsteoBites next week when we chat with Dr. Thank you. Thanks so much, Dr. Yang. Thanks, Walker.
Thank you.